In this video, we're going to look at the structure of amino acids and also how they form into peptide chains. Amino acids are the building block for proteins. There are 20 amino acids in human proteins, and we're going to look at the general structure of amino acids and then how those 20 different amino acids come about. So what I have here on the screen is just the general structure of an amino acid, and you can tell from the name some of the structure that is present. And so you have amino and acid. And if we look down here at the structure, here we have an amino group. And on the other side, we have a carboxylic acid functional group. So you have both the amino and the acid functional group, which is where it gets its name as amino acid. In the middle, in between there, we have what's called an alpha carbon. And this alpha carbon is a chiral carbon in all of the amino acids except for one. But in this alpha carbon, and let me change colors for my pen, you have the amino groups, you have a hydrogen, the carboxylic acid, and then this R group at the top. And the R group is what can differ from one amino acid to the other. And we'll see examples of that as we go along. But depending on what the R group is, defines it as a certain amino acid. So in all of the amino acids, the R groups are going to be different. And all of the amino acids are chiral except for glycine that has an H for an R group. So that's the only one that's not chiral. Otherwise, all of the amino acids are chiral. The active form, the form that is found in human proteins, are L amino acids. Now, the amino acids, the 20 different amino acids that are found in human proteins can be divided or grouped into different groups based on what the side chains are. And so we're going to look at some of these different groups. And on this slide, I have listed both nonpolar and polar amino acids. These are not all of them. I've just given you a couple of examples of each of them. And so I'm going to circle in red this side chain. This is the R group for each of these. Now you'll notice on the two I just circled, those are non-polar, and those side chains just contain carbons and hydrogens, which are very non-polar, have non-polar bonds, and so that's what makes them non-polar amino acids. Now on the polar amino acids, here we have serine, and it has a hydroxyl group at the end of it. Now that OH bond is a very polar bond, and so that makes it a polar amino acid. And for cysteine, you have a sulfur hydryl group, and so it makes it a polar bond as well. Cysteine is also an important amino acid, and we'll see it when we look at proteins in a different video, because that sulfhydryl group will form disulfide bonds, and so we'll look at that later, but just kind of keep cysteine in mind there. The parts on all four of these amino acids that I did not circle, if you will look at them, they are exactly the same. They have the amino group, that alpha carbon bonded to a hydrogen, and the carboxyl group. And so that's the conserved region, or the part of the amino acid that is same for all of the 20 amino acids. The other two groups of amino acids that we have are polar acidic amino acids and then polar basic amino acids. And so I've given you a couple examples for these, starting with this first one. I'm going to circle the R group, the side chain here, and you can see on it, it has that carboxylic acid functional group, and so that's our organic acid, so that's why it is classified as an acidic amino acid, and that functional group is very polar, which is why it's a polar acidic amino acid. And then lysine over here is our polar basic amino acid. I'm going to circle this, and you can tell at the top you have that amine group, which makes it a basic amino acid. But then the parts that I did not circle, again, have that conserved region. It's the same between all of them. When amino acids come together, they will form what's known as a peptide bond to start to form a protein. And so I'm going to just draw out how that is. So we're going to start with drawing an amino acid. Here's my alpha carbon with the hydrogen bonding to a carboxyl group here. Off of my alpha carbon, I'll just put this as a CH3. So this is alanine. It doesn't matter which amino acid it is because the, see, that has a plus sign, because the reaction takes place between that conserved region. And so they're all going to bond the same no matter which amino acid it is. And the side chain is just kind of hanging off from that. So here's my other amino acid. And this is a dehydration reaction. So when this forms a peptide bond, when these two amino acids bond together, they lose water. And so the reaction takes place between the carboxyl group on one amino acid and the amine group on another amino acid. Here's my alpha carbon, hydrogen, R group. And the carbonyl is still there, and now it loses its oxygen. It bonds to the nitrogen from the other amino acid. There's the other alpha carbon, my R group, and the carboxyl group. And it lost, if you'll notice, it lost 2 hydrogens and an oxygen. So it lost a water. And in the middle that connects both of those, so right here is our peptide bond. And that is what holds 2 amino acids together in the chain. And you can see that on one side you have a free amino group, on the other side you have a free carboxyl group. And so that carboxyl group is able to bond to the amino group of a different amino acid and form another peptide bond, and you can get long chains from that. Let's look at a peptide. So I'm going to draw out for you a peptide chain. So we're going to start with the nitrogen here for one. There's my alpha carbon, and I'm going to write the side chain, the part that can differ from one amino acid to the other, as just an R group. So that's R1. Here's my nitrogen, alpha carbon. We'll say this is R2. Carbonyl bonding to a nitrogen, bonding to an alpha carbon, R3, and we'll do one more. There's R4. Okay, so here is a 4 amino acid peptide chain. And I'm just going to kind of define a few things for you. I've written out the side chains, the things that can differ from one amino acid to the other as R groups. So R1, R2, R3, R4. They could be the same, they could be different, but these are referred to as side chains. This terminology, what I'm defining for you, becomes important when we talk about protein structure, which is a different video, but it will become important. This part that I'm circling right now is the conserved region. This is the thing, the peptide bonds, the part of all of the amino acids that are the same. This is referred to as the backbone, the conserved region in our polypeptide chain here. And this side, you notice we have a free amino group. This is called the N terminal, N for the nitrogen that's there. And the other side with the free carboxyl group is the C terminal. And this becomes important when we're looking at the structure of proteins and what order those amino acids are bonding in. It's the free N terminal or the free C terminal, and it gives you the order that they are bonded in. And so these terms that I've listed here are important to remember when you are learning about the different structures of proteins, and we'll talk about that in the video about protein structure.